I opened this meeting by looking back 200 years, so now let's look into the future. Some 50 years back I wrote an article in Nature entitled Microscopic Blind Spots. And I said how odd it is that the public happily recognise the DNA spiral, the rings around Saturn, the craters on the moon, had no idea what a living cell looks like. Well, since then, it's got a lot worse. Those of you who read the journal The Microscope, I hope everybody at this meeting does, you may have seen my article entitled Farewell Reality, Hello Jelly Beans. Cells are being shown as simplified blobs. Since then, it's got far worse. With the advent of AI, people are now inventing creatures that have never existed. We're entering the era of the mythical microbe. I have published examples from the many reference books that showed cartoons of bacteria that didn't exist in life. They were crude and to any microscopist unrecognisable. I said they were quite unlike any organisms that actually exist. But worse was to come. Whenever you see a report online these days about living cells, there are always these crude jelly bean cartoons. It's truly quite remarkable. Living cells are beautiful. Yet a cartoon like this, a drawing like this, is meant to represent what living cells are like. And of course the public will take it at face value. If this has been published by somebody who puts themselves forward as an expert on life, then you'll believe that that's what living cells are like. But of course the majority of cells are nothing like that at all. Look at this report. The extracellular matrix is the key to longevity. That sounds good and scholarly and academic, but once again, a childlike drawing of nothing remotely like living cells. Professor Yoshinori published studies of autophagy some years ago, and the graphic that accompanied this post a few weeks ago is nothing but crude jelly beans. Look at this from an article on the pancreas. Perfect, beautiful, voluptuous jelly bean cells, and nothing like any cells that you'll ever find in the pancreas. Let's look online and do a search for living cells. And as you scroll down through Google, you see endless graphics. They're virtually all of those jelly bean models. It's hard to find anything close to a living cell, no matter where you look. The BBC posted a site, Nature Wildlife, so that you could scroll down the various forms of life, amphibians, lampreys, cephalopods, and right down to the clitellata, the earthworms. No sign of any microbes. Microbes are the most important regulators of the living world that exist. And yet, in all the extensive archives of the BBC, there is nothing at all on a microbe. The corporations seem almost blind to the fact.
When I first started out, there was so much of the natural world we were unable to film. The cameras, the lighting, the size of the equipment, all meant that the extremely small, the very large, the ultra-fast or the infinitesimally slow were beyond our reach. In the last 30 years, things have changed out of all recognition. We can now film everything, from the giants of the world's oceans to the smallest invertebrates.
The leafhopper is a member of the family Psychedelidae, but it has 5 million cells. And my dear and much respected friend David Attenborough says, this is as small as we normally go, and it's absolutely true. The last time that I showed the microscopic world to a broadcast audience was some 35 years ago, when TV presenter Carol Vorderman came to see the sights that we all know.
You can see the nucleus, which of course is the...
She was captivated by the sight of the protozoan Peronema.
Head at the front. That's the flagellum, which is...
Unforgettable sights.
It is quite amazing, the beautiful things that you can see under the light.
Yes, it is, and at the end of it.
Yet it's a sight the television audience never gets to see. When the BBC did their TV series about the cell, I was excited to think that this would be a chance to reveal the majesty of the microbe, the complexity of living cells, their sheer voluptuous, vital beauty. But in fact, almost everything we saw was a computer graphic, with about as much life as cinders on a dead barbecue.
We are cells. Every time we breathe, we move, we think, cells do the work for us. Like all biologists, the more I find out about them, the more they amaze me.
The story of the cell is the most powerful story in science. It's the theory of everything.
And just look at the graphics that they showed to illustrate what living cells were like. Blobs of jelly, formless, uninteresting, unlifelike, and unreal. My spirits rose when my great friend Professor George McGavin told me he was going to be doing a series, Miniature Britain, which would have the microscope in it. He was off to the sea and promised to be looking closely at marine phytoplankton. Well, this is what we saw.
And then what we can do is we can just wash that out into a cup, and we can collect that to look at under the microscope later.
To actually see them, we're going to have to push our microscope camera to its very limits. What I really want to see, Emilian, is the phytoplankton.
Maybe we can zoom in on this one right here.
Oh, that's nice.
Yeah, they look like some diatoms. Oh, there he is.
Oh, that is beautiful. Fantastic.
And in fact, these final sequences are 3 video micrographs of particles of dirt. The videos laid one upon the other. This is the kind of view of marine phytoplankton that I thought George would be showing everybody. Look at the life, the intricacy, the sheer beauty of microbes at work. I've often proposed to the BBC that we should make a programme about living cells. Had a discussion with somebody from the Natural History unit called Max last year, and then their head of programming came back to me with a letter saying, no, we haven't had a strong enough interest to proceed. Other people have been making documentary films purporting to show living cells.
If scientists succeed in uncovering all the secrets of the cell, it can be a tremendous benefit for all humanity. Cell research is crucial to understanding how life works. The possibilities are enormous as scientists continue learning more and more about these building blocks of life. For the cell's life is part of our life.
Once again, they're unbelievably crude and simplified. These aren't cells. This is computer graphics. I cannot tell you how disappointed I was to see this crude distortion of the magnificence of life. Now that AI has taken off, misrepresentation is everywhere. Let's have a look at this Euglena cell busying itself in a pond and photographed by my late friend Jeremy Pickett-Heaps. You see the red eye spot. You see the flagellum. This is an exciting organism full of life, full of vivacity. And here is an amoeba. Every part of that cell has a purpose. Every bit of it knows what it's supposed to do. We regard them as the simplest form of life, but come now, this is an organism composed primarily of water-soluble components. The very fact that it doesn't just disperse into the pond water is a miracle in itself. And here is a group of paramecium, busying themselves in a protozoan culture, photographed by Dr. Bob Burnham in Canada. But now let's see where the educators come in. MTISH Tech International. They have a site purporting to turn words into visuals, artificial intelligence for smart teachers. So let's see what they made of it all.
The diagram shows various cellular organisms. Let's see them in action. The organelles pulsagelib waves in nodonsin. The paramecias and cilia beat in waves for propulsions.
Euglena has its silky body labelled flagellum, and I see the bacterium has pills. I do hope it's feeling better now. I wonder what clysosome is, and what was it he said?
The organelles pulsagelib waves in nodonsin.
It's ignorant from start to finish.
Prokaryotic bacteria are simple cells with a nucleoid and a flagellum for movement. An amoeba moves and eats using its flexible pseudopodia, which help it swim.
There's a cell what? Eucgmas, a plast cell that waves at you. Euglena with its spiky cell wall labelled flagellum, and a starfruit in a plant cell to end. Here is an amoeba, exploring its environment under the microscope. Put it under high power and you get some impression of the complexity of what's going on inside. Amoebae are remarkable organisms. Some of them, as I'm sure you know, build shells in which they live. Amoebae are unique. This is the MTISH-TECH view of an amoeba. I have to say it doesn't bear a resemblance to any amoeba known to me. Experiment 3D. They have produced the most remarkable graphics. They're nonsensical, they're non-existent, they're inaccurate, they're misleading, but they're done with incredible technical skill. Here's a paramecium sculling along through the pond. What a remarkable organism that is. See how briskly its cilia beat in unison to propel it wherever it wishes to go. The sculpturing of its cell wall and the radiating paths that lead down to the contractile vacuole, its equivalent of a kidney. And here is a rotifer, producing vortices in the water which, with luck, will sweep food into its gullet. Well, this is what Experiment 3D makes of it. And their rotifer looks like a power tool, and nothing at all like life. If you watch the way that an amoeba moves, you can see that it has a permanently changing morphology. You can recognize an amoeba very often by the shape of its pseudopodia. Yet, paradoxically, they're never the same shape from one moment to the next. Now let's see what Experiment 3D makes of it. No amoeba has ever acted like this. The clarity of the image, the sheer technical skill is extraordinary. But their amoeba has 4 appendages and goes lumbering about like a baby dinosaur. Just look at that again. One of the most common of the freshwater chlorophyte algae is Chlamydomonas. And here is a colony of bemused Chlamydomonas cells. This is what Arcadia science made of it. A simplified diagram, but I suppose it serves a purpose. But this is MTISH-TECH's view. I cannot begin to imagine how anybody could relate that to anything to do with life. Now let's see how they explained reproduction by the means of spores.
Spore formation begins within a protective structure. The spores develop and are released into the environment. They are then dispersed to start a new cycle. Spore formation begins in the spore mother cell. Through meiosis, it divides into multiple cells. These develop into spores with protective walls. They mature and prepare for dispersal. Spore fungi produce reproductive cells within specialized...
You see, the graphics are superb. They're so professional, they're beautifully done. It's just that they bear no relationship whatever to life. They're totally misleading. They're a digital lie. So let us consider mitosis, the essential process that gives life to all multicellular creatures. If you look at a time-lapse movie, this is speeded up about 20 times. Cell division normally takes about 1 hour. And here are animal cells. You can see the chromosomes drawn apart to form 2 new nuclei. And now that that's happened, of course, the chromosomes disappear. People don't seem to realize that chromosomes only appear when the nucleus is going to divide. So let's look closely at the chromosomes. And you see how there's a lot of jostling going on. Chromosomes are actively moving when they're about to divide. They sort themselves out, they get into order, and then the spindle fibers pull them apart to form 2 daughter nuclei. Now, Jeremy Pickett-Heaps had his team produce this very neat graphic. It is a cartoon, it is a computer representation, but it clearly shows how the spindles operate in drawing the chromosomes apart. GCSE biology, that's the examination in Britain for 16-year-olds, I'm afraid has simplified it to a quite remarkable extent. Just watch this.
In the second stage of the cell cycle, mitosis takes place. One set of chromosomes is pulled to each end of the cell. The nucleus also divides.
The cognitosite has a version of their own. At least, they feature drawings of something that looks vaguely like chromosomes.
All 46 of the cell's chromosomes line up along the center of the cell. At this point, fibers from either side of the cell attach to their respective half of each chromosome. These are called cell fibers, and they pull the arms of the chromosomes to opposite sides of the cell. We call these two sides poles. As you can see, this breaks the chromosomes in half, into two separate arms. This happens to all 46 of the chromosomes.
These accounts may be limited, they may be crude, but they're certainly not as bizarre as the BBC. Spending hundreds of thousands of pounds on a programme where the cell is just reduced to nothing. The population is taught that living cells are simple little things, which cell biologists are on the point of creating for themselves, and this appalling BBC graphic of a blob of jelly falling in half, like droplets on a window, perpetuates the myth. In fact, the spindle is birefringent and it shows up with dramatic clarity under polarised light. Here you can visualise how the spindle forms as it attaches to chromosomes and draws them apart. Let's have a look at the graphic version that Pickett Heaps and his team produced to show mitosis. At the start of mitosis, two centrosomes form against the nucleus. The chromosomes suddenly condense and become visible. The centrosomes then produce spindle fibres, some of which from opposite poles overlap and slide against each other. They will help to push the poles apart and maintain the shape of the spindle. As the centrosomes ready themselves to migrate to opposite poles, the nuclear envelope disperses and disappears. The spindle fibres can now attach to the kinetochores located at the centromere on each chromosome. There is much oscillation of the chromosomes as they jostle into position. Such is the clumsy choreography of mitosis. Yet this is what MTish Tech comes up with. A confusing graphic that bears no relationship to anything that happens in life. And their graphic explaining mitosis could be the worst I have ever seen.
During prophase, chromosomes condense and the nuclear membrane breaks down. In metaphase, the chromosomes align at the center of the cell. Anaphase follows, with sister chromatids being pulled to opposite poles. Finally, in telophase and tituletinesis, two new cells are formed.
Nothing like that exists in nature. No matter how tolerant we might be of exaggeration, or simplification, distortion, graphic artifact, this is a curious cacophony of confusion. So we have seen the elegant reality of microscopic life. The graceful swimming of a living paramecium with its coordinated cilia, the baffling ballet of mitosis, the quiet ingenuity of living cells, contrasted with the wildly distorted monsters flooding the internet. These crazy cartoons blur truth and fantasy for the student and compromise the public understanding of the science. Yet the actual microscopic world needs no embellishment. It is far more beautiful and astonishing than any digital fantasy. The public are now being bombarded online with endless videos of microbes that don't exist. The past was bad enough. I hate to think for our future.